Hi semua! So in today's video, we're going to be doing a simple osmosis experiment with potatoes. But before that, allow me to explain the meaning of osmosis. Osmosis is the passive movement of water molecules from a region with lower concentration of solute to a region with higher concentration of solute through a partially permeable membrane until an equilibrium is reached. The main purpose of this experiment is to investigate the effect of different salt concentrations on the potato cells through osmosis process. So, without wasting any time, let's jump right into the experiment. You might be wondering, what are the materials used in this experiment? Well, I'm gonna tell you right now. First and foremost, of course, we're going to need potatoes. We're going to cut these potatoes into small potato strips later. Next, we need water, a measuring cup, salt, a chopping board, a knife, a ruler, a scale, a cork borer or a strong plastic straw, and 3 cups. Now, let's take a look at the procedure of the osmosis experiment using potatoes. First, you will need a cork borer to get your potato strips. In this experiment, I don't have a cork borer, so I'm just using a hard straw. I'm pushing the hard straw into the potato and taking a long stick to push the potato strips out of the straw. We will need 9 potato strips. After that, make sure all of the potato strips are same in length. For this experiment, I'll be cutting my potato strips into 5 centimeters. Measure the potato strips with a ruler accurately first before cutting them. Set the potato strips aside and we will now be preparing the solutions. Measure 200 ml of water using the measuring cup and fill each cup with 200 ml of water. Take out your mini scale and add salt to the scale until it reads 5 grams. Since my scale is basically broken, I'll be using 1 tablespoon of salt. Add the salt that we have just measured into the second cup and we have just made a salt solution of 5 grams per 200 milliliters. In this experiment, we need 3 solutions. 0 grams per 200 ml, which is just pure water, 5 grams per 200 ml, and lastly 10 grams per 200 ml. Repeat this step until all the solutions are prepared. After that, put 3 potato strips in each of the cups and make sure the potato strips are immersed completely. Start the timer and leave them where they are for 30 minutes. After 30 minutes, we have to take out and measure all of the potato strips and measure them as accurately as possible. To the nearest millimeter is good for this experiment. As you're working through the potato strips, you'll probably notice that the ones immersed in more concentrated solutions are more soft, while the ones immersed in less concentrated solutions are more turgid. Make sure to write down each value you measure as you go along. And lastly, tabulate the data. From the data obtained, we have tabulated the initial and final length of the potato strips. We have also calculated and tabulated the average change in length of each potato strips in different concentration of solutions. So, from the average of change in length of the potato strips, we can deduce that when the potato strip is put into a less concentrated solution, or also known as dilute solution, the potato strip becomes turgid. Therefore, the conclusion is, the lower the concentration of solute, the more turgid and bigger the size of the potato strips. After doing this experiment, I've learned that water molecules will always move from a region of high water potential to a region with lower water potential. Hence, if a potato strip is immersed in a hypertonic solution, the potato strip will become soft and plasmolized. On the other hand, if a potato strip is immersed in hypotonic solution, the potato strip will become turgid. That's all from me. I hope you had fun watching my video and learned something new. Thank you. Bye bye.